Some of the animals you think are extinct are on their way back to life right now. We've unlocked the secrets of DNA, and there are laboratories rewriting the code of the dead. We didn't just watch these animals die. We decided to bring them back. 10 animals the world gave up on that are currently being reconstructed in clean rooms, in test tubes, in the dark. A few have already taken their first breaths after we'd buried them. And the last one? Scientists are planning to release it into the wild in just a few years.
Number 10. Gastric brooding frog. Some people will tell you that once an animal is gone, its unique biological secrets are lost forever. They're not entirely wrong, but in the mid-1980s, a tiny, bizarre amphibian vanished from the rainforests of Australia, and scientists have been obsessed with it ever since. The gastric brooding frog had a reproductive method that sounds like science fiction. The female would swallow her fertilized eggs, completely shut down her stomach's hydrochloric acid production, safely brood her young inside her digestive tract, and then give birth directly through her mouth. Under the Lazarus Project, researchers used dead somatic tissue that had been preserved in a conventional home freezer for decades. Using somatic cell nuclear transfer, they deactivated the nuclei of living eggs from a distant frog relative and replaced them with the frozen gastric brooding frog DNA. Here's what's strange. The eggs actually revived, began dividing, and grew into early-stage embryos. Though the cells currently stall before morphing into living tadpoles, the team is actively troubleshooting the genetic switches to cross the finish line. But if a frog turning its stomach into a nursery sounds bizarre, the next animal looks like a zebra that simply ran out of ink.
Number 9. Quagga. Deep in the historic plains of South Africa, a creature that looked like a hybrid of a zebra and a horse once roamed in vast herds. It had stripes on its head and neck, but its hindquarters were solid brown. By 1883, the last one died in an Amsterdam zoo, and the quagga was declared extinct. Here's where the science gets interesting. DNA testing from preserved museum pelts proved the quagga wasn't a distinct equine species at all. It was actually a geographic subspecies of the common plains zebra that had simply lost its rear stripes over time. Under the Quagga project in South Africa, researchers realized they didn't need high-tech gene splicing to bring it back. They just needed selective breeding. Over 4 decades of pairing plains zebras with reduced striping has yielded animals virtually indistinguishable from original quagga specimens. These animals, called row quaggas, are now successfully breeding true across large reserves in South Africa's Western Cape. They are reclaiming their original grazing niche, proving that sometimes extinction is just skin deep. But breeding the stripes off a zebra is a slow process. The next creature was a massive wild ox that ruled ancient European forests, and scientists had to rebuild it from the ground up.
Number 8. Aurochs. Picture a wild ox standing 6 feet tall at the shoulder, with massive, forward-pointing horns and an aggressive temperament that made it the king of the ancient European wilderness. This was the aurochs, the direct ancestor of all modern domestic cattle. It ruled the forests for thousands of years until the very last one died in Poland's Yaktorow forest in 1627. Because the aurochs is the forefather of modern cattle, its complete DNA profile still exists, scattered across primitive, unselected European breeds like the Italian Maremma and Spanish Sayagesa. Under the Tauros program and Rewilding Europe, scientists are using a process called backbreeding. By selectively pairing cattle that carry high percentages of primitive aurochs traits across multiple generations, they have successfully bred Tauros cattle. These animals mirror the massive size, horn shape, and coat color of the extinct wild ox. Today, wild, self-sufficient herds are already grazing across European nature reserves, with official reintroductions landing in the Scottish Highlands.
They act as heavy landscape engineers, clearing thick brushwood to create diverse, sunlit forest pastures. Recreating a wild bull through selective breeding is one thing, but the next animal on our list actually achieved literal de-extinction, if only for a few minutes.
Number 7. Pyrenean Ibex. In January 2000, the last living Pyrenean Ibex, locally known as the bucardo, was found dead in the Pyrenees Mountains, her skull crushed by a falling tree. Her name was Celia, and with her death, an entire subspecies of alpine goat was wiped off the map. But before she died, scientists took skin biopsies and deep froze them in liquid nitrogen. In 2003, Spanish researchers used somatic cell nuclear transfer to insert Celia's DNA into the eggs of domestic goats, implanting them into surrogates. Against all odds, a live bucardo kid was born, but the triumph was short-lived. The clone died just 7 minutes after birth due to a severe lung deformity. While no active cloning attempts are currently underway, Celia's intact genetic lines remain cryopreserved, waiting for reproductive technology to catch up. A clone that lived for 7 minutes proved the concept was possible. But while scientists cloned an ibex from frozen tissue, the next creature requires sequencing DNA from bones that have been buried for hundreds of years. Number 6. South Island giant moa. Before humans arrived in New Zealand, the islands were ruled not by mammals, but by birds. The undisputed king was the South Island giant moa, a flightless herbivore that stood up to 12 feet tall and weighed nearly 500 pounds. Without any native mammalian predators, they thrived until Maori settlers arrived and hunted them to extinction around 1400 AD. Now, Colossal Biosciences, in collaboration with the Ngai Tahu Research Center, is working to bring the giant back. Using high-throughput paleogenomic sequencing, scientists are extracting DNA from fossilized moa toe bones and mapping it against its closest living evolutionary relatives, like the flightless tinamo and the emu. Backed by high-profile investors, including filmmaker Peter Jackson, researchers are charting the genetic blueprint of this lost giant. The primary technical hurdle is scaling up reproductive tech, as giant moa eggs were vastly larger than ostrich eggs. But if a giant bird egg feels like a massive biological challenge, the next species once filled the sky by the billions, and its disappearance permanently changed the forests of North America.
Number 5. Passenger pigeon. In the 19th century, the passenger pigeon was the most abundant bird in North America, and perhaps the world. They traveled in colossal, dark flocks of billions that would literally block out the sun for days. But relentless commercial hunting and habitat destruction wiped them out with terrifying speed. In 1914, the last representative of the species, Martha, died alone in her cage at the Cincinnati Zoo. The initiative Revive and Restore is working to change that. By recovering passenger pigeon DNA from taxidermy specimens, scientists are editing the genome of the closely related band-tailed pigeon. The team has already successfully mapped the genome and isolated the specific mutations that gave the passenger pigeon its distinct social flocking behavior, bright coloring, and lightning-fast breeding cycles. In the wild, their massive flocks once created natural forest disturbances, breaking branches and leaving thick layers of nutrient-rich droppings that regenerated hardwood forests. That entire ecological cycle collapsed when the birds vanished, and bringing them back could be the key to restoring our forests. But while passenger pigeons are being engineered to save the woods, the next bird is the most famous symbol of extinction itself.
Number 4. Dodo Bird. On the isolated island of Mauritius, a large, flightless, and entirely fearless bird lived in peace. Because it had no natural predators, the dodo bird had no reason to fear the European sailors who arrived in the 17th century. Within less than 100 years of human contact, the dodo was gone, becoming the universal symbol of human-caused extinction. Now, scientists are attempting a highly complex process called primordial germ cell transfer to bring it back.
Because editing avian eggs is notoriously difficult due to the hard shell and yolk structure, scientists must isolate the embryonic cells that form sperm and eggs, edit them with dodo DNA, and implant them into a host bird, in this case, the Nicobar pigeon. In an avian bioengineering breakthrough, researchers developed custom 3D printed artificial eggshells containing specialized internal lattice structures. They have successfully hatched healthy live chicks within these artificial environments, clearing the path to hatch the first engineered dodo proxies. If successful, the dodo will return to Mauritius as a keystone seed disperser, reviving native forests that have stalled out since the bird vanished. An artificial eggshell could bring back a flightless bird from an island. But the next creature is a legendary marsupial predator whose unique biology makes it much easier to grow in a lab.
Number 3. Tasmanian Tiger. In 1936, the last Tasmanian tiger, or thylacine, died of neglect in a Hobart zoo. It was a unique marsupial predator with a stiff gait, a jaw that could open to an astonishing angle, and tiger-like stripes running down its back. Farmers blamed them for sheep losses, leading to a government-funded eradication campaign that wiped them out. Today, Colossal Biosciences and the University of Melbourne's Tiger Lab are using a well-preserved joey kept in a museum jar to sequence an ultra-high-quality genome. They plan to edit this sequence into the genes of the fat-tailed dunnart, a mouse-sized marsupial. Here is the strange part. Marsupials are actually highly advantageous for de-extinction. Their gestation periods last only a few weeks, and the tiny, underdeveloped offspring finish growing outside the womb, inside a pouch. This allows scientists to run reproductive experiments exponentially faster than they can with placental mammals. Reintroducing this apex marsupial to Tasmania could control overpopulated non-native species and restore balance to the ecosystem. But while the thylacine is still in the planning stages, the next creature on our list is an Ice Age predator that is already walking the earth.
Number 2. Direwolf. During the last Ice Age, the direwolf was one of the most formidable apex predators in North America. Built heavier and more muscular than modern gray wolves, they hunted massive prey across the frozen landscape before vanishing roughly 10,000 years ago. For a long time, people assumed they were just slightly larger gray wolves, but a genetic study proved direwolves split from other canids 5.7 million years ago, making them very distant relatives. Because of this, scientists at Colossal Biosciences had to engineer the traits entirely from scratch. Using a 13,000-year-old tooth found in Ohio to map the ancient genome, they made 20 targeted edits across 14 genes in gray wolf blood cells to replicate direwolf morphology. This project has already produced living results. 3 genetically modified pups were successfully born via surrogate hound mixes, Romulus and Remus, and later Khaleesi. They currently reside in a secure, high-tech, 2,000-acre ecological preserve, growing 20% faster and larger than standard gray wolves. By studying these resurrected predators, scientists hope to develop genetic tools to rescue critically endangered living canids, like the North American red wolf. A pack of engineered direwolves running through a private preserve sounds like science fiction. But the final animal on our list is the ultimate giant of the Ice Age, and scientists are building it to save the planet from itself.
Number 1. Woolly mammoth. Imagine an animal standing 11 feet tall, weighing 6 tons, and covered in a thick coat of shaggy hair with curved tusks longer than a grown man is tall. The woolly mammoth is the definitive icon of the Ice Age. Most of them vanished around 10,000 years ago, though a tiny, isolated population survived on Wrangel Island until about 4,000 years ago. Now, Dr. George Church and the team at Colossal Biosciences are using CRISPR gene editing to bring it back. By mapping mammoth DNA extracted from permafrost fossils, scientists are editing those specific genes into the genome of the Asian elephant, which shares a 99.6% genetic match. Researchers have already identified and target edited roughly 25 of the 85 core genes responsible for mammoth traits, such as thick fat layers, small ears to prevent heat loss, and shaggy hair. Mammoth embryos are currently being optimized for artificial womb gestation, with a live hybrid birth targeted in the coming years. But this isn't just a science experiment. Reintroducing mammoths to the Arctic tundra has an ecological purpose to combat climate change. By trampling heavy snow and punching through insulating moss layers, these giants will allow freezing air to reach deep underground, preventing the melting of carbon-trapping permafrost.
We didn't just watch them die. We are actively choosing to bring them back. The labs are running, the DNA is being rewritten, and the surrogates are waiting. So maybe the question isn't whether we can resurrect the dead. It's how the world will change when they finally walk among us again. Subscribe and don't look away from the wild.